Okay, can somebody confirm you can hear me?
I can hear you.
I can hear you.
I should be sharing my screen. Today is January 11th, and we're going to be talking about Lab 01, the ubiquity of microorganisms. You should also realize that you must get the worksheet for Lab 00 in by 8 a.m. Friday, January 12th, or you will be dropped from this class. This is the only attendance I'm going to be taking in this class. The worksheet for Lab 01 will not be due until 11:59 p.m. Saturday, January 13th. Let me remind you, you must also complete the plagiarism quiz by this Saturday, 11:59 p.m. You must obtain 100% on this plagiarism quiz. If you do not get 100%, you can take the quiz again. And rarely do I ever have a student needing to take the quiz more than twice. Rarely students even take it twice. Let me also add that you should read the infectious disease research project before our next lab, because I'm going to be discussing the infectious disease project in our next lab. So the infectious disease project will be the paper you're supposed to read is the project instructions. You can find it on campus. Let me show it to you. Scroll down to Lab 1. And it's not in there, so let's go down to Lab 2. There it is, the Infectious Disease Research Project. It's a 100-point project. You're going to be making a research project on an infectious disease. And you can read about it here, and I'll discuss it next lab. Any questions about anything? If not, let's go to the lab. We're going to be talking about the ubiquity of microorganisms, Lab 01. I don't think I have that open. Lab Module 1, Ubiquity of Microorganisms. For, I think, every lab, there will be a little bit of reading out of your lecture textbook. So you should read for this lab pages 157 to 162, covering culture media. And if you have a different edition than the most recent edition of the tutorial textbook that's made for Clark College, you'll have to figure out what pages to read, but it's going to be covering culture media, and it will be around those pages. Then read a little bit about obtaining pure cultures, and this is only a page, and then the culture media is what, about 4 or 5 pages. There are some supplemental materials in this lab, video clips. The learning objectives for this lab are: 1, know that microorganisms are almost everywhere. 2, describe the purpose of culture media. 3, describe what colony abundance is and colony morphology. And then 4, be able to define the following terms: inoculation, ubiquity of microorganisms, what's something that's sterile, isolation, incubation, in vitro, culture media. And when we have culture media, there are different types. There can be a broth or a liquid culture media, and then there can be a solid culture media, and those are usually solidified by adding agar to the liquid culture media. And you can grow these in a slant. Let me see if I can draw that. I'll talk about it later. Or you can have the solid media in either a slant or a deep. I'll show that later. What does fastidious mean? What's a non-selective media? What's ubiquitous mean? And what's a colony? Any questions about any of that? All right. Microorganisms are organisms that are too small to be seen with the naked eye. This does include viruses and prions, which are not organisms, and they're included in the group even though they aren't alive, or at least some people don't consider them alive. We'll also include helminths, worms in our study, even though the adult worms are not microscopic. However, their infective stages of the worms are microscopic. And then worms also cause diseases. The phrase ubiquity of microorganisms refers to the concept that microorganisms are almost everywhere. Sterile, something that's sterile means that it's free of living cells. In the microbiology lab, if you were to go in to a real microbiology lab, you would be given sterile media, and how it is removed of all living cells is they autoclave it. There are some other things you could do to sterilize the media, and we will talk about different ways you can sterilize things. Like when we're talking about the goggles, we sterilize them with UV light or exposure to UV light. In this lab, we're going to investigate the abundance and diversity of microorganisms in common environments. We'll also learn some basic lab protocols. A culture medium, also called a growth medium, is a sterile laboratory preparation that contains a variety of nutrients that are required for growing microorganisms in the lab, and that is in vitro. Any question about any of that? As I already stated, culture medias may be a liquid. We call those broths, and they're usually put in a test tube, because if you put a broth in a plate, it can easily spill out like an agar petri plate. We usually put broths in a test tube. You can add agar to a liquid media to make it a solid media. Agar has no nutritional value. It's a gelatinous substance abstracted from seaweed. We can grow agar. Let me see if I've got that down below. Yeah, right here. We usually put solid media in a petri dish, but you can grow it in a test tube, meaning put the solid media in a test tube. Here is a test tube of liquid media, a broth, and that is solid media, and this we call a slant. For obvious reasons, the media is in a slant. And this we call a deep, where the agar is just straight across. And these are both solid medias. Any question about any of that? All right, here's a little video you can watch on preparing core plates. Note how the culture media needs to be sterilized prior to use. And here's a question for you. Why must a culture medium be sterile prior to it being used? Anyone have a guess? All right.
To make sure no microorganisms contaminate it?
Yes. If you have more than one microorganism in whatever you're studying, And if you get a positive result from whatever you're studying, you don't know which organism created the positive result if you have more than one. And so microbiologists usually study one organism at a time. And they do this by using a sterile medium, as well as only putting in one species into the media. All right, today we're going to talk about general purpose media. These are non-selective media. They're usually complex in composition, and they support the growth of most non-fastidious organisms. They usually support the growth by added peptones or yeast or beef extracts. There are three general purpose media that we need to talk about. But first, let me say that a fastidious organism is one that does not grow easily. In order to get a fastidious organism to grow, you need to add specific additives to the medium. A fastidious organism will not grow in a general purpose media. Any question about any of that? An example of a fastidious organism is Streptococcus pyogenes, which causes strep throat and many other human diseases. Most of these diseases are before my time, so it's something like, I think, scarlet fever and rheumatoid fever, which were very common in America before the development of antibiotics. But usually when somebody gets Streptococcus pyogenes, they have strep throat and they have this sore throat for a long period of time, like a month, and then they go in to see the doctor and they realize they have strep throat and they put the patient on antibiotics. And so the disease never progresses further. At least normally in first world countries, that's the way it happens now. But strep throat used to cause a whole bunch of other diseases before the introduction of antibiotics. And my point is Streptococcus pyogenes is a fastidious organism. It will not grow on a general purpose media. The first general purpose media I'm going to mention is number three, nutrient agar. All it has is, let's see if I can remember, beef extract, sodium chloride, and one other ingredient. I don't remember what it is. We'll talk about it later in the lab. It might be peptone, which is just digested proteins. Tryptic soy agar is a second general purpose media, and I guess nutrient agar can be abbreviated NA, tryptic soy agar, TSA. And then nutrient agar with yeast extract is the same as nutrient agar. You just add yeast extracts. It's a little richer. And that's abbreviated NA. Any question about any of that? These are the three most common media you run into in a microbiology lab. All right, whenever you're growing things in the lab, the tube or the dish should be completely labeled prior to its use with the following 4 or 5 items included on the dish or tube. You must include your name, the date, the type of medium like NA, the inoculum, that's what you're growing. If you know what it is, you put it, like E. coli. If you don't, then you do something like air exposure. You don't need to say growth from air exposure, just say air exposure on it. And then you can put the incubation temperature you grow it at, and that's optional. It is not required. But all the other 4 items are required on your container you're growing your organisms in. And there you can see a dish labeled with the person's name, what's growing in the dish, the date the dish was streaked out, the media, TSA, and then optionally the temperature you grow it at. Whenever you write on a Petri dish that you're growing organisms in, you should write on the bottom. You know, I didn't mention that to my other class. So let me make a note of that. Why we write on the bottom? I probably do mention it in another lab, but we put things on the bottom of the plate because that is where the agar and the cells will be. If you're right on the lid, and then the lid gets separated from the base of the plate, you won't know what's growing in the plate. And I once had a person in a lab, it was actually next door to my lab, they had a whole tray full of different plates, and each one of them had written on it what was growing on the plate. And as she was carrying the whole tray, she tripped, and all the plates went on the floor, and the lids got separated from the bases. And then nobody knew what was what. And so the whole experiment had to be tossed. And so you should write everything on the base. Make a note of that. And I need to mention that to my other class. You should also write on the edge of the bottom of the plate, because if you write here, you'll have a hard time seeing the growth. So you write along the edge. An inoculation is the placement or transfer of a microbial specimen into or on a sterile culture medium. So when you put growth in your plate or your microbial cells in your plate, we call that an inoculation. Incubation is then the placement of the culture media containing the growth in an appropriate physical environment for growing the microbes for a period of time. And usually we incubate our dishes in an incubator. This is a picture of an incubator, a rather fancy one. You shut the door and then it heats up. It's actually heated up now, but it's going to be cooled because the door is open. Yeah, all incubators do that. They have a set temperature you can grow them at, and you can change the temperature. And it adds humidity. What you do is you usually put water in a container in the bottom of the incubator, and that adds water to or humidity to the incubator that prevents the plate from drying out rapidly. Fancy incubators can also control the level of CO2 the cells will be exposed to and the level of oxygen the cells will be exposed to. In our labs, we don't have fancy incubators. All we do is we have incubators that control the temperature, the humidity, and then obviously the time. Any question about any of that? All right, when you're growing plates in an incubator, let me empty this. This will be my pretend plate. When you put a plate in the incubator will warm the plate up, and then the moisture in the agar will evaporate, and it'll condense on the lid. And then a drop will form on the lid, and then the drop will eventually drip down onto the plate. that usually does not contaminate the plate, although it does make the risk of contamination greater because I don't know if you can see this, but the edge, this part of the edge of the plate is not sterile. The lid inside surface is sterile. So if the drip forms there and then drops, it shouldn't be non-sterile. But if the drip were to come down the side and get in the bottom of the plate, it could contaminate the plate. So the main reason is mainly that the drop of water will drop on the bacteria we have growing and then disperse them. So instead of having individual colonies, you get all the cells growing together. So to prevent that from happening, the water dripping down onto your growth, we put the Petri dishes in the incubator upside down. Now, if the moisture were to condense or evaporate and then condense, it'll evaporate on the surface, which is the agar. And the agar will wick the moisture so that there will never be a drop forming. And even if a drop were to form, it would then drip on the lid, which who cares because we don't have any bacteria there. So we always put our Petri dishes in the incubator upside down to prevent the dripping of drops of water down onto our growth. Any question about any of that? All right. So once you get the bacteria to grow on a Petri dish with solid media, the growth will form visible bumps on the agar surface. Each of these will be a colony. Bacterial colonies tend to have a rather smooth textured surface, and that differs from a mold or fungal colony. Excuse me here. So these are bacteria colonies. You can see they have a very distinct edge, very smooth edge. And that differs from a fungal colony. Let me blow that up a little bit, where the edges is kind of hairy or fuzzy. And then this one, you can see it a little better if I shrink it down. That one looks hairy or fuzzy there, at least in the dark part. In the white part, it's kind of hard to see. It's a little bit hairy. I have to blow it up really large though. There, you can sort of see it fuzzy there. So you can make some distinction just by looking at the colonies. A colony is made up of tens to hundreds of millions of cells. Each colony, to be visible, even if it's a tiny speck, has at least 1 million cells. All of those cells in the colony may be derived from a single one cell. That one cell divides into two cells into four, four into eight, eight into 16, 16 into 32, etc. Up until you get 1 million cells. And if you get 1 million cells, you can then visibly see the colony, although 1 million will be just a speck. Now, not all colonies are formed from a single cell. A colony could be formed from 2 cells or even a clump of cells. And when you look at colonies, you can't determine, is that colony formed from a single cell, 2 cells, or a clump of cells? Sometimes you can kind of guess, like the largest colonies, like that one there is much larger than all of these. It's even larger than that one. And probably this colony here was formed by a clump of cells, and these were probably formed by single cells, but you can't always tell, like that one is in between these and that. And so, like I said, you can't always tell. Any question about any of that? Oh, excuse me here.
Is that something that you'll want us to know?
Know that colonies are made of one cell, two cells, or a clump of cells?
Yes, and I'm sorry, I mean, do you want us to be able to identify that or to be able
Uh,
to guess?
no, you don't really need to be able to guess that, but you should know that that can happen.
Okay.
Um, like I said, it isn't always the case. Like, let me show you this picture here. Whenever you have colonies which are closer together, they're usually sharing the resources, so they tend to be a little smaller than a colony which is further out, like this colony and that colony. So these colonies have more resources, so they'll be a little bit bigger. But that one is pretty close to that one, and it's much larger than all the others. So my guess is that one is formed by a clump, but you can't always determine.
Okay.
Okay.
Thank you.
And so, no, I won't ask you to try and determine that. All right, here's a little video you can watch on binary fission. You should watch that before you begin working directly on the lab. I think I already talked about that. Let me shrink this down a little. All right. In theory, one individual cell leads to the formation of one colony, yet a colony may be formed by two or more cells, including a clump of cells. You should know that colonies are not visible in a broth culture. In a liquid culture, all of the cells will mix together and you get no colony formation. You can sometimes see obvious visible colony morphological differences between different species of bacteria. These we call colony characteristics. Colonies have a particular shape. They can be circular, rhizoid, irregular, filamentous, or spindle in shape. They can have a specific margin. The entire margin is just a smooth edge. The margin is the edge of the colony. So a circular colony will have an entire edge. A irregular colony, and maybe a filamentous, will have undulate or lobate edge. And I'll never ask you, is this colony undulate, lobate, or curled? Because these are all pretty similar. But I would ask you a question like, is that margin entire, or is it undulate, lobate, or curled? Do you understand? So I'm not going to ask you to separate these three, but I would ask you to separate these three from this one. Okay? And then the same with rhizoid and filamentous. I would never ask you, is this colony rhizoid or filamentous? But I could ask you, is this colony rhizoid / filamentous, or is it entire? Any question about that? All right. Colonies also have an elevation. You can have a colony which is flat. You can have a colony which is raised. You can have a colony which is convex. You can have a colony which is pulvinate, and that's just extreme convex. I would never ask you, is this colony convex or pulvinate? Because to tell you the truth, I would not be able to separate those two. And then there's umbunate, and it kind of looks like a mountain. To see the elevation of a colony, you really have to take the lid off the plate and then look at the colony from its edge. Because when you look straight down, you cannot see or determine the elevation. Let me show you a picture of that. Oh, this is a little better here. Nope, this is better because it's got it labeled here. They're telling you that colony A here is pole venate. When I look at colony A, I cannot see any elevation. I can't tell you if that's flat, fulvinate, umbunate, or whatever. And that's because we're looking at it on top, looking straight down, and you can't see the elevation. And then colony B is supposed to be a flat colony. I can't tell that either. And then colony C is a convex colony. Like I said, I can't distinguish that one from that one or that one. Any question there? So to see the elevation, you really need to look at the colony from its side. The size of the colony is, if it's punctiform, it's a pinpricked. If it's small, that means it's about the size of a pen or a somewhat dull pencil. Moderate is in between the size of the eraser of the pencil and then the sharpened edge of the pencil. Something in between that. Anything that's over the size or the same size as the eraser on a pencil would be large. Colonies can be smooth or rough in their appearance. They can be glistening or shiny, or they can be dull, which is also their appearance. Colonies can be pigmented. If a colony is pigmented, then it has a color, like purple, red, or yellow. If it's non-pigmented, the colony, the colony lacks color. It can be a white colony, an off-white colony, a tan-colored, or a cream-colored colony, but that's a non-pigmented colony. Colonies also have an opaque, optical property. Colonies can be opaque. Most colonies are opaque. A colony can be translucent. Only some colonies are translucent. Translucent means you can see through the colony, but you can't see through it very well. Transparent means the colony is transparent, like a drop of water. Very few colonies are transparent. Coming down here, this colony is opaque. This colony is opaque. That colony is opaque. See this big colony here? Let me blow this up. I'm not talking about that part there, but this part there, this is translucent. That one there is probably translucent as well. Any question about any of that? You'll note I'm not showing you any transparent ones because none of these colonies are transparent. All right. Two terms used with colonies are abundance and diversity. Abundance is simply the total number of colonies on an agar plate. To get the abundance, all you have to do is count them. So on this one, I'm not going to count all of them, but I'll count them from here up. It's going to be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 in this region of the plate. The abundance in this region of the plate is 13. Now, when I'm asking for abundance, you can be close, meaning if I were to say, what's the abundance of this region of the plate? If you were to tell me a number close to 13, I will say that's good enough. And that would be something like 10 to 16. But if you go beyond 16 or less than 10, I'm going to say that's not close enough. And obviously it will depend on how many colonies are on the plate. Like right here, I would say the abundance is something like 40 to 50. It may be higher, like 60, but I'm going to guess 40 to 50. And I'm not going to actually count them. There's too many to count, at least for me right now. But that's what you do to get the abundance. You simply count them. Total number of colonies. Diversity is the number of different kinds of colonies on an agar plate. Coming back to this picture here, just because it's easier to work with than that picture there. Well, there's two different fungal colonies, so that's 2. And then these are probably all the same. So that's 3, and that's going to be the same too. But that one's different, so that's 4. This one's different, so that's 5. That one's different, so that's 6. And that one is probably different. So there's probably 7 different, at least 7 different colony types growing on this plate. And so I would say the diversity is 7 or maybe 8 if you count maybe those small ones there. So 7 or 8 is the diversity. And if you're close to that, I'll say that's close enough. Like 5 would be close enough. A 9 would be close enough. But if you're off, like you told me 3, I'm going to say, no, that's not good enough. Okay. Any question about any of that? So in the abundance and diversity, as long as you're close enough, it'll be correct. You don't have to be exactly the same numbers as I got. And I would hate, I'd hate counting all of the colonies on this plate and then trying to distinguish the different types. There's a large number of different types on this plate. The diversity is very large. Here's a real world practice example. In this image, what is the abundance of wildcats in the picture? Can somebody answer that?
7.
Yeah, 2 lions and 1, 2, 3, 4, 5 cheetah. So that's abundance of 7 wildcats. And what's the diversity of cat species in this picture?
3.
3. What's the third?
The cheetahs.
There's only 1 species of cheetah. These are all cheetahs. And those are both lions. So there's only a diversity of 2 cat species. Okay. So the diversity would be 2. All right. I was hoping that would be, everyone would get that correct, but this was a really simple example of abundance and diversity. All right, know the terms of this lab. An interesting picture of somebody's fingerprint or handprint. And let's move to the worksheet. Remember, the worksheet has to be turned in by Saturday of week 1. That's this Saturday at 11:59 p.m. Remember, worksheet 00 for lab 00 must be turned in by 8 a.m. on Friday. And I just got an email saying I need to get my information in who's attending the class by 5 p.m. Friday. But you guys don't have that time limit, I do. And I do need a little bit of time between 8 a.m. and the time I need to get it in. So we're going to stick with 8 a.m. on Friday. All right, lab module 1, the ubiquity of microorganisms. In procedure 1, you're going to be answering what is in the air you breathe. Below is a nutrient agar plate that was exposed to the air and then incubated for 24 to 48 hours at 30 degrees Celsius in an incubator. Procedure 1A, question 1A, answer, what is the colony abundance on this plate? All you need to do is count the colonies. And then procedure 1B, what is the colony diversity on the plate? This is a little trickier. You have to distinguish between the different species on the plate. And I'll tell you that those two, as well as that one, are probably the same. But these two are different. Okay? And there are some other different ones that's clearly different too. So give me the number of colony diversity on the plate. And you don't have to be exact, you just need to be close. And then procedure 1C, what step would you take to reduce airborne contamination of an sterile agar plate while you are inoculating it? I get students all the time telling me, put it in the lid, tape the lid on shut, and then put it in the incubator upside down, and that's their answer. That is not something you're doing while you are inoculating the plate. Okay, so what's something you can do while you are inoculating the plate to reduce airborne contamination falling into the plate? And that is actually discussed in the lab module, but it's pretty obvious once you think about it. So don't tell me something you can do after you're inoculating it, and then don't tell me something you can do before you inoculate the plate. Like I've occasionally had students say, always use a plate that's sterile, has no contamination in it, or something like that. That's something that happens before you inoculate the plate. Procedure 2, what gets on your arms when you put your arms on the lab bench? What we're going to do is sample the lab bench for microbes before you apply the disinfectant, shown here, and then after you disinfect the lab bench. And you can see the differences between the two here. And then the first question, A, did table disinfection work? It's just yes or no, but you can't explain it if you wish. And then question B, what is the purpose of allowing the table disinfectant to air dry? Or in other words, why do we air dry the disinfectant? Meaning when we put the disinfectant on the table, we allow it to air dry. All right, any question about that? Procedure 3, what is on your hands? Plates A are the plates of a person's hands. 1, 2, 3, 4 fingers. 1, 2, 3, 4 fingers. And then the growth coming from their hand before they wash their hands. And plates B is the growth from the fingers after they wash their hands and dry their hands. The first question to ask, number 1 for procedures is 3. Procedure 3, number 1, is does hand washing work? Once again, that's just a yes or no, or you can explain it if you wish. And then question 2, suggest one reason why hand washing did not result in the complete removal of microbes. Hint, it may have something to do with the normal microbiota of the skin. If you obtain an answer following research, like online or in your book, please provide a brief citation of your information source. This is a thinking question. It cannot be found directly in the lab module. And even when you do research, you may have to think a little bit about it. Procedure 4. When you kiss your significant other, what exactly are you giving him or her? Meaning what is on your lips and what is on your tongue. So I guess we're talking about French kissing. So this is somebody's plate after they expose the plate to their lip. I'm going to assume that's the bottom lip and that's the top lip. And this is the plate after it's been exposed to the person's tongue. Please note that I've added a note there for the smear applying on the tongue plate. So you can read that note. And then answer the question, which plate has higher colony abundance, the lip plate or the tongue plate? And then question B, why do you think that this is the case, even though the lips and the tongue are nearby each other, and you probably often lick your lips with your tongue? So what might cause the observed difference in colony abundance between the lips and the tongue? There are actually 3 easy answers when you think about this. And I've had brilliant students come up with like 5 different answers. I am going to require that you supply at least 2 different answers for there being differences between the colony abundance on the lips or the tongue. Any question about that? So for full credit here, you have to supply 2 answers. 2 reasons why the colony abundance on one plate is greater than the other plate, meaning one of them, the lip or the tongue, is higher than the other. And then why might that be? And you must supply 2 answers for why. And then C, why was the tongue plate incubated at a higher temperature than the lip plate? And once you think about that, that'll be obvious too. Procedure 5 are microorganisms in the soil. What we've done is we take some soil, I diluted the soil in water, and then take a small sample and put that on the plate. And then the first question, procedure 5A, what is the colony abundance on this plate? Is it even possible to accurately answer this question? Please explain your answer. And then question B, is there greater abundance of colonies from the soil compared to the lip plate? Here. And I've had people state, well, the lip plate has small colonies, so there's more than the soil colonies because the soil colonies are all large. And I'm going to tell you right there, the lip plate does have some large colonies. That's a large one, that's a large one, that's a large one, that's a large one. The majority are small though. And then the soil plate, let me blow this up, does have some small colonies. Some small ones right there, a whole bunch of small ones there, a whole bunch of small ones right there. There are some, like this one, and that one, which are very large, but not all of them are large. I would say less than half of them are large, and at least half of them are small. And many times you can't see the small colonies in certain regions of the plate. You can only see them in certain regions of the plate. And that's because the growth on this plane is so heavy right there, you can't determine how many colonies are right there. All right, any questions about this lab? If not, I will be here until something like 4:20 to help you answer questions. Let me see what time the lab ends. Oh, the lab's supposed to end at 4. You know, I don't think that's right. I'll have to look into that. I'll be here until 4:20, okay? All right, get started on the lab. If you have any questions, that's why I'm staying here. I will be here until the last student logs out.
Thank you for your time.
All right,
Thank you.
you're welcome.
Thank you.
Talking about these labs being, you said you get to do them twice if you don't do well on them like the first time.
Yeah, they'll be graded twice. The first time
Okay.
you get the grading, you will have about a week to turn in the lab a second time, and it will be graded for 100%.
Okay, thank you.
All right.